Welcome back, my dear students. Today we are going to explore an essential cellular organelle of the GRL system or of the cell, that is the lysosome, often called as the cell's digestive system. These tiny enzyme-packed vesicles break down waste, recycle cellular materials and help maintain overall cell health. So let's dive into its structure, functions and significance in a clear and simple way to understand how lysosomes keep our cells clean and efficient. Discovery of lysosome, it was discovered by Christian D. Dubé in 1955 using a subcellular fractionation technique. It was observed under electron microscope by Novikov in 1956, who also coined the term lysosome. Occurrence. Lysosome is basically present in all animal cells except RBCs. It is highly abundant in leukocytes, macrophages, Kupffer cells, that is cells with phagocytic activity and activity. In plants and fungi, vacuoles perform similar functions. Origin or biogenesis of the lysosome, it is formed by the coordinated action of endoplasmic reticulum, endosome and Golgi complex, that is the GRL system. The enzymes within lysosome, that is the precursor of the enzymes, are synthesized on rough endoplasmic reticulum. Structure of lysosomes, shape and size. Lysosomes are basically globular or granular structures and its size range between 0.2 to 5.5 micron. They do not have a fixed shape. Membrane structure. Each lysosome is surrounded by a single lipid membrane. The membrane isolates powerful digestive enzymes from the rest of the cell. Membrane is impermeable to the substrates of the enzyme, preventing accidental digestion of the cell. Enzyme contain 50 types of hydrolytic or digestive enzymes stored as crystalline or semi-crystalline granules. Different lysosomes contain different sets of enzymes which are following, that is acid phosphatase, sulfatases, proteases, peptidases, nucleases, lipases and carbohydrates. These enzymes are collectively called acid hydrolases. Acidic environment, enzymes work very fast at pH 4 to 5, that is acidic. Acidity is maintained by pumping H plus ions or protons into the lysosome. Suicidal bag of the cell, lysosomes contain strong digestive enzymes that can break down all biomolecules. If the membrane breaks, enzymes leak out, cell undergoes lysis or autolysis, hence they are termed as suicidal bag. Membrane stability, labilizer, that is the substances that weaken the lysosomal membrane or enzyme escape, cell components get digested. Stabilizers, that is the substances that strengthen the membrane, prevent leakage and protect the cell from autolysis. Polymorphism of lysosome. Lysosome exists in multiple forms within a cell depending on their function at stage. This phenomena is called as polymorphism. There are basically four types of lysosome. Primary lysosomes, they are newly formed vesicles that bud off from Golgi apparatus. They are smaller in size. They contain inactive hydrolytic enzymes such as stored as granules. They become fully functional only after fusing with endosome or phagosome. Secondary lysosome or heterophagosome or digestive vacuoles, they formed by the fusion of primary lysosome and phagosome, that is the food containing vesicle, site of active digestion of engulfed food particles. Digested nutrients diffuse into the cytoplasm. Undigested materials remain inside and later from residual bodies. Residual bodies or tertiary lysosomes contain only waste left after digestion, usually move to the cell surface and release debris by exocytosis or Autophagy, that is external digestion. If waste is not expelled, these bodies accumulate, causing storage disease like Pompey's disease, Hurler's syndrome, Tay-Sachs disease, hepatitis, polynephritis. Lipofuction granules are actually residual bodies. So autophagic vacuoles or autophagosomes or autolysosomes, they are formed when primary lysosomes are damaged or worn out organelles. Before fusion, the organelle is wrapped by membranes derived from endoplasmic reticulum. It carry out autophagy, that is self-digestion of cellular components. It help in removal of old, injured or non-functional organelles. Apoptosis or programmed cell death, recycling of useful molecules such as used for new synthesis, removing internal obstructions within the cell, providing nutrients during starvation by breaking down cellular reserves. Because of their role in digestion and recycling, lysosomes are called disposal unit or recycling centers. This is the functions of lysosomes: intracellular digestion, extracellular digestion, defense mechanism, autophagy, removal of obstructions, mobilization of reserves, intracellular scavenging, sperm lysine, disposal of useless cells, role in storage disease, thyroxine formation, cell division, genetic effects, carcinogenesis, leukocyte granules, bone remodeling. So, my dear students, today we learned how lysosomes, the cell's digestive sacs, plays a vital role in breaking down waste, recycling cellular material, and protecting the cell from harmful substances, from maintaining cellular health to supporting metabolism and defense. Lysosomes are truly essential for proper cell function. If you enjoyed learning about these amazing organelles, don't forget to like, share, and subscribe for more exciting cell biology classes. See you in the next class. Thank you.